Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cell nucleus. 100 languages. HeLa cells stained for nuclear DNA with the blue fluorescent UHs dye. The central and rightmost cells are in interface, thus their entire nuclei are labeled. On the left, a cell is going through mitosis and its DNA has condensed. Cell biology. Animal cell diagram. Components of a typical animal cell. 1 nucleolus. 2 nucleus. 3 ribosome, dots as part of 5. 4 vesicle. The cell nucleus, from Latin nucleus or nucleus kernel, seed, pl, nuclei, is a membrane-bound organelle found in eukaryotic cells. Eukaryotic cells usually have a single nucleus, but a few cell types, such as mammalian red blood cells, have no nuclei, and a few others including osteoclasts have many. The main structures making up the nucleus are the nuclear envelope, a double membrane that encloses the entire organelle and isolates its contents from the cellular cytoplasm, and the nuclear matrix, a network within the nucleus that adds mechanical support. The cell nucleus contains nearly all of the cell's genome. Nuclear DNA is often organized into multiple chromosomes, long strands of DNA dotted with various proteins, such as histones, that protect and organize the DNA. The genes within these chromosomes are structured in such a way to promote cell function. The nucleus maintains the integrity of genes and controls the activities of the cell by regulating gene expression. Because the nuclear envelope is impermeable to large molecules, nuclear pores are required to regulate nuclear transport of molecules across the envelope. The pores cross both nuclear membranes, providing a channel through which larger molecules must be actively transported by carrier proteins while allowing free movement of small molecules and ions. Movement. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 5 rough endoplasmic reticulum. 6 Golgi apparatus, or Golgi body. 7 cytoskeleton. 8 smooth endoplasmic reticulum. 9 mitochondrion. 10 vacuole. 11 cytosol, fluid that contains organelles, with which comprises cytoplasm. 12 lysosome. 13 centrosome. 14 cell membrane. A mouse fibroblast nucleus, in which DNA is stained blue. The distinct chromosome. Territories of chromosome 2, red, and chromosome 9, green, are stained with fluorescent in situ. Hybridization of large molecules such as proteins and RNA through the pores is required for both gene expression and the maintenance of chromosomes. Although the interior of the nucleus does not contain any membrane-bound subcompartments, a number of nuclear bodies exist, made up of unique proteins, RNA molecules, and particular parts of the chromosomes. The best known of these is the nucleolus, involved in the assembly of ribosomes. Chromosomes. Main article, chromosome. Further information, Nuclear organization. The cell nucleus contains the majority of the cell's genetic material, in the form of multiple linear DNA molecules organized into structures called chromosomes. Each human cell contains roughly 2 meters of DNA. 405 during most of the cell cycle. These are organized in a DNA protein complex known as chromatin, and during cell division the chromatin can be seen to form the well-defined chromosomes familiar from a karyotype. A small fraction of the cell's genes are located instead in the mitochondria. 438. There are two types of chromatin. Euchromatin is the less compact DNA form, and contains genes that are frequently expressed by the cell. 2. The other type, heterochromatin, is the more compact form, and contains DNA that is infrequently transcribed. This structure is further categorized into facultative heterochromatin, consisting of genes that are organized as heterochromatin only in certain cell types or at certain stages of development, and constitutive heterochromatin that consists of chromosome structural components such as telomeres and centromeres. 3. During interphase the chromatin organizes itself into discrete individual patches, 4, called chromosome territories. 5. Active genes, which are generally found in the euchromatic region of the chromosome, tend to be located towards the chromosome's territory boundary. 6. Antibodies to certain types of chromatin organization, in particular, nucleosomes, have been associated with a number of autoimmune diseases, such as systemic lupus erythematosus. 7. These are known as antinuclear antibodies, ANA, and have also been observed in concert with multiple sclerosis as part of general immune system dysfunction. 8. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Diagram of the nucleus showing the ribosome studied outer nuclear membrane. Nuclear pores, DNA, complexed as chromatin, and the nucleolus. A cross-section of a nuclear pore on the surface of the nuclear envelope. 1. Other diagram labels show, 2, the outer ring, 3, spokes, 4, basket, and 5, filaments. Nuclear structures and landmarks. Further information, nuclear equivalence. The nucleus contains nearly all of the cell's DNA, surrounded by a network of fibrous intermediate filaments called the nuclear matrix, and is enveloped in a double membrane called the nuclear envelope. The nuclear envelope separates the fluid inside the nucleus, called the nucleoplasm, from the rest of the cell. The size of the nucleus is correlated to the size of the cell, and this ratio is reported across a range of cell types and species. 9. In eukaryotes, the nucleus in many cells typically occupies 10% of the cell volume. 10. 178. The nucleus is the largest organelle in animal cells. 11. 12. In human cells, the diameter of the nucleus is approximately 6 micrometers, m. 10. 179. Nuclear envelope and pores. Main articles, nuclear envelope and nuclear pore. The nuclear envelope consists of two membranes, an inner and an outer nuclear membrane, perforated by nuclear pores. 10. 649 together, These membranes serve to separate the cell's genetic material from the rest of the cell contents, and allow the nucleus to maintain an environment distinct from the rest of the cell. Despite their close apposition around much of the nucleus, the two membranes differ substantially in shape and contents. The inner membrane surrounds the nuclear content, providing its defining edge. 14 Embedded within the inner membrane, various proteins bind the intermediate filaments that give the nucleus its structure. 649 The outer membrane encloses the inner membrane, and is continuous with the adjacent endoplasmic reticulum membrane. 649 As part of the endoplasmic reticulum membrane, the outer nuclear membrane is studded with ribosomes that are actively translating proteins across membrane. 649 The space between the two membranes is called the perinuclear space, and is continuous with the endoplasmic reticulum lumen. 649 In a mammalian nuclear envelope there are between 3,000 and 4,000 nuclear pore complexes, NPCs, perforating the envelope. 650 Each NPC contains an 8-fold symmetric ring-shaped structure at. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A position where the inner and outer membranes fuse. 12. The number of NPCs can vary considerably across cell types. Small glial cells only have about a few hundred, with large Purkinje cells having around 20,000. 650 The NPC provides selective transport of molecules between the nucleoplasm and the cytosol. 13. The nuclear pore complex is composed of approximately 30 different proteins known as nucleoporins. 649 The pores are about 60–80 million Daltons in molecular weight and consist of around 50, in yeast, to several hundred proteins, in vertebrates. 6224 The pores are 100 nm in total diameter, however, the gap through which molecules freely diffuse is only about 9 nm wide, due to the presence of regulatory systems within the center of the pore. This size selectively allows the passage of small water-soluble molecules, while preventing larger molecules, such as nucleic acids and larger proteins, from inappropriately entering or exiting the nucleus. These large molecules must be actively transported into the nucleus. Instead, attached to the ring is a structure called the nuclear basket that extends into the nucleoplasm, and a series of filamentous extensions that reach into the cytoplasm. Both structures serve to mediate binding to nuclear transport proteins. 1, 50910. Most proteins, ribosomal subunits, and some RNAs are transported through the pore complexes in a process mediated by a family of transport factors known as karyophorins. Those karyophorins that mediate movement into the nucleus are also called importins, whereas those that mediate movement out of the nucleus are called exportins. Most karyophorins interact directly with their cargo, although some use adapter proteins. 14. Steroid hormones such as cortisol and aldosterone, as well as other small lipid-soluble molecules involved in intercellular signaling, can diffuse through the cell membrane and into the cytoplasm, where they bind nuclear receptor proteins that are trafficked into the nucleus. There they serve as transcription factors when bound to their ligand. In the absence of a ligand, many such receptors function as histone destylases that repress gene expression. 488. Nuclear lamina. Main article, nuclear lamina. In animal cells, two networks of intermediate filaments provide the nucleus with mechanical support. The nuclear lamina forms an organized meshwork on the internal face of the envelope, while less organized support is provided on the cytosolic face of the envelope. Both systems provide structural support for the nuclear envelope and anchoring sites for chromosomes and nuclear pores. 15. The nuclear lamina is composed mostly of lamin proteins. Like all proteins, lamins are synthesized in the cytoplasm and later transported to the nucleus interior, where they are assembled before being incorporated into the existing network of nuclear lamina. 16, 17. Lamins found on the cytosolic face of the membrane, such as emerin and nesprin, bind to the cytoskeleton to provide structural support. Lamins are also found inside the nucleoplasm where they form another regular structure, known as the nucleoplasmic veil. 18, 19, that is visible using fluorescence microscopy. The actual function of the veil is not clear, although it is excluded from the nucleolus and is present during interphase. 20. Lamin. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. An electron micrograph of a cell nucleus, showing the darkly stained nucleolus. Structures that make up the veil, such as LEM3, bind chromatin and disrupting their structure inhibits transcription of protein-coding genes. 21. Like the components of other intermediate filaments, the lamin monomer contains an alpha helical domain used by two monomers to coil around each other, forming a dimer structure called a coiled coil. Two of these dimer structures then join side by side, in an antiparallel arrangement, to form a tetramer called a protofilament. 8 of these protofilaments form a lateral arrangement that is twisted to form a rope-like filament. These filaments can be assembled or disassembled in a dynamic manner, meaning that changes in the length of the filament depend on the competing rates of filament addition and removal. 15. Mutations in lamin genes leading to defects in filament assembly cause a group of rare genetic disorders known as laminopathies. The most notable laminopathy is the family of diseases known as progeria, which causes the appearance of premature aging in those with the condition. The exact mechanism by which the associated biochemical changes give rise to the aged phenotype is not well understood. 22. Nucleolus. Main article, nucleolus. Further information, nuclear bodies. The nucleolus is the largest of the discrete densely stained membraneless structures known as nuclear bodies found in the nucleus. It forms around tandem repeats of rDNA, DNA coding for ribosomal RNA, rRNA. These regions are called nucleolar organizer regions, NOR. The main roles of the nucleolus are to synthesize rRNA and assemble ribosomes. The structural cohesion of the nucleolus depends on its activity, as ribosomal assembly in the nucleolus results in the transient association of nucleolar components, facilitating further ribosomal assembly, and hence further association. This model is supported by observations that Inactivation of rDNA results in intermingling of nucleolar structures. 23. In the first step of ribosome assembly, a protein called RNA polymerase I transcribes rDNA, which forms a large pre-RNA precursor. This is cleaved into two large RNA subunits 5.8s and 28s, and a small RNA subunit 18s. 10. 328, 24. The transcription, post-transcriptional processing, and assembly of RNA occurs in the nucleolus, aided by small nucleolar RNA, snRNA, molecules, some of which are derived from spliced introns from messenger RNAs encoding genes related to ribosomal function. The assembled ribosomal subunits are the largest structures passed through the nuclear pores. 1. 526. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. When observed under the electron microscope, the nucleolus can be seen to consist of three distinguishable regions: the innermost fibrillar centers, FCs, surrounded by the dense fibrillar component, DFC, that contains fibrillarin and nucleolin, which in turn is bordered by the granular component, GC, that contains the protein nucleophosmin. Transcription of the rDNA occurs either in the FC or at the FC-DFC boundary, and, therefore, when rDNA transcription in the cell is increased, more FCs are detected. Most of the cleavage and modification of RNAs occurs in the DFC, while the latter steps involving protein assembly onto the ribosomal subunits occur in the GC. 24. Splicing speckles. Speckles are subnuclear structures that are enriched in pre-messenger RNA splicing factors and are located in the interchromatin regions of the nucleoplasm of mammalian cells. 25. At the fluorescence microscope level they appear as irregular, punctate structures, which vary in size and shape, and when examined by electron microscopy they are seen as clusters of interchromatin granules. Speckles are dynamic structures, and both their protein and RNA protein components can cycle continuously between speckles and other nuclear locations, including active transcription sites. Speckles can work with 53P as enhancers of gene activity to directly enhance the activity of certain genes. Moreover, speckle-associating and non-associating 53P gene targets are functionally distinct. 26. Studies on the composition, structure and behavior of speckles have provided a model for understanding the functional compartmentalization of the nucleus and the organization of the gene expression machinery. 27. Splicing snRNPs, 28, 29, and other splicing proteins necessary for pre-RNA processing. 27. Because of a cell's changing requirements, the composition and location of these bodies changes according to mRNA transcription and regulation via phosphorylation of specific proteins. 30. The splicing speckles are also known as nuclear speckles, nuclear specks, splicing factor compartments, SF compartments, interchromatin granule clusters, IGCs, and B snoposomes. 31. B snoposomes are found in the amphibian oocyte nuclei and in Drosophila melanogaster embryos. B snoposomes appear alone or attached to the cagal bodies in the electron micrographs of the amphibian nuclei. 32. While nuclear speckles were originally thought to be storage sites for the splicing factors, 33. A more recent study demonstrated that organizing genes and pre-mRNA substrates near speckles increases the kinetic efficiency of pre-mRNA splicing, ultimately boosting protein levels by modulation of splicing. 34. Cagal bodies and gems. A nucleus typically contains between 1 and 10 compact structures called cagal bodies or coiled bodies, CB, whose diameter measures between 0.2 micrometer and 2.0 micrometers depending on the cell type and species. 35. When seen under an electron microscope, they resemble balls of tangled thread. 36. And are dense foci of distribution for the protein coilin. 37. CBs are involved in a number of different roles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Cagal body. Subnuclear structure sizes. Structure name. Structure. Diameter. Ref. Cagal bodies 0.22.0 m. 35. Clastosomes 0.20.5 m. 40. Pica 5 micrometers. 36. PML bodies 0.21.0 m. 41. Paraspeckles 0.51.0 m. 42. Speckles 20-25 nm. 36. Relating to RNA processing, specifically small nucleolar RNA, snRNA, and small nuclear RNA, snRNA, maturation, and histone mRNA modification. 35. Similar to cagal bodies are Gemini of cagal bodies, or gems, whose name is derived from the Gemini constellation in reference to their close twin relationship with CBs. Gems are similar in size and shape to CBs, and in fact are virtually indistinguishable under the microscope. 37. Unlike CBs, gems do not contain small nuclear ribonucleoproteins, snRNPs, but do contain a protein called survival of motor neuron, SMN, whose function relates to snRNP biogenesis. Gems are believed to assist CBs in snRNP biogenesis. 38. Though it has also been suggested from microscopy evidence that CBs and gems are different manifestations of the same structure. 37. Later ultrastructural studies have shown gems to be twins of cagal bodies, with the difference being in the coilin component. Cagal bodies are SMN positive and coilin positive, and gems are SMN positive and coilin negative. 39. Other nuclear bodies. Main article, Nuclear bodies. Beyond the nuclear bodies first described by Santiago Ramon y Cagal above, for example, nucleolus, nuclear speckles, cagal bodies, the nucleus contains a number of other nuclear bodies. These include polymorphic interphase karyosomal association, pica, promyelocytic leukemia, PML, bodies, and paraspeckles. Although little is known about a number of these domains, they are significant in that they show that the nucleoplasm is not a uniform mixture, but rather contains organized functional subdomains. 41. Other subnuclear structures appear as part of abnormal disease processes. For example, the presence of small intranuclear rods has been reported in some cases of Nemarlin myopathy. This condition typically results from mutations in actin, and the rods themselves consist of mutant actin as well as other cytoskeletal proteins. 43. Pica and PTF domains. Pica domains, or polymorphic interface karyosomal associations, were first described in microscopy studies in 1991. Their function remains unclear, though they were not thought to be associated with active DNA replication, transcription, or RNA processing. 44. They have been found to explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Often associate with discrete domains defined by dense localization of the transcription factor PTF, which promotes transcription of small nuclear RNA, snRNA. 45. PML nuclear bodies. Promyelocytic leukemia protein, PML nuclear bodies, are spherical bodies found scattered throughout the nucleoplasm, measuring around 0.11.0 m. They are known by a number of other names, including nuclear domain 10, ND10, CREMA bodies, and PML oncogenic domains. 46. PML nuclear bodies are named after one of their major components, the promyelocytic leukemia protein, PML. They are often seen in the nucleus in association with cagal bodies and cleavage bodies. 41. PML/mice, which are unable to create PML nuclear bodies, develop normally without obvious ill effects, showing that PML nuclear bodies are not required for most essential biological processes. 47. Paraspeckles. Main article, paraspeckle. Discovered by Fox et al. In 2002, paraspeckles are irregularly shaped compartments in the interchromatin space of the nucleus. 48. First documented in HeLa cells, where there are generally 10-30 per nucleus. 49. Paraspeckles are now known to also exist in all human primary cells, transformed cell lines, and tissue sections. 50. Their name is derived from their distribution in the nucleus. The para is short for parallel and the speckles refers to the splicing speckles to which they are always in close proximity. 49. Paraspeckles sequester nuclear proteins and RNA and thus appear to function as a molecular sponge. 51. That is involved in the regulation of gene expression. 52. Furthermore, paraspeckles are dynamic structures that are altered in response to changes in cellular metabolic activity. They are transcription dependent. 48. And in the absence of RNA Pol II transcription, the paraspeckle disappears and all of its associated protein components, PSP1, P54NRB, PSP2, CFI, M68, and PSF, form a crescent-shaped perinucleolar cap in the nucleolus. This phenomenon is demonstrated during the cell cycle. In the cell cycle, paraspeckles are present during interphase and during all of mitosis, except for telophase. During telophase, when the two daughter nuclei are formed, there is no RNA Pol II transcription, so the protein components instead form a perinucleolar cap. 50. Perichromatin fibrils. Perichromatin fibrils are visible only under electron microscope. They are located next to the transcriptionally active chromatin and are hypothesized to be the sites of active premna processing. 33. Clastosomes. Clastosomes are small nuclear bodies, 0.20.5 m, described as having a thick ring shape due to the peripheral capsule around these bodies. 40. This name is derived from the Greek klastos, broken. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Answer: body. 40. Clastosomes are not typically present in normal cells, making them hard to detect. They form under high proteolytic conditions within the nucleus and degrade once there is a decrease in activity or if cells are treated with proteasome inhibitors. 53. The scarcity of clastosomes in cells indicates that they are not required for proteasome function. 54. Osmotic stress has also been shown to cause the formation of clastosomes. 55. These nuclear bodies contain catalytic and regulatory subunits of the proteasome and its substrates, indicating that clastosomes are sites for degrading proteins. 54. Function. The nucleus provides a site for genetic transcription that is segregated from the location of translation in the cytoplasm, allowing levels of gene regulation that are not available to prokaryotes. The main function of the cell nucleus is to control gene expression and mediate the replication of DNA during the cell cycle. 171. Cell compartmentalization. The nuclear envelope allows control of the nuclear contents, and separates them from the rest of the cytoplasm where necessary. This is important for controlling processes on either side of the nuclear membrane. In most cases where a cytoplasmic process needs to be restricted, a key participant is removed to the nucleus, where it interacts with transcription factors to downregulate the production of certain enzymes in the pathway. This regulatory mechanism occurs in the case of glycolysis, a cellular pathway for breaking down glucose to produce energy. Hexokinase is an enzyme responsible for the first step of glycolysis, forming glucose 6-phosphate from glucose. At high concentrations of fructose 6-phosphate, a molecule made later from glucose 6-phosphate, a regulator protein removes hexokinase to the nucleus. 56, where it forms a transcriptional repressor complex with nuclear proteins to reduce the expression of genes involved in glycolysis. 57. In order to control which genes are being transcribed, the cell separates some transcription factor proteins responsible for regulating gene expression from physical access to the DNA until they are activated by other signaling pathways. This prevents even low levels of inappropriate gene expression. For example, in the case of NFB-controlled genes, which are involved in most inflammatory responses, transcription is induced in response to a signal pathway such as that initiated by the signaling molecule TNF, binds to a cell membrane receptor, resulting in the recruitment of signaling proteins, and eventually activating the transcription factor NFB. A nuclear localization signal on the NFB protein allows it to be transported through the nuclear pore and into the nucleus, where it stimulates the transcription of the target genes. 15. The compartmentalization allows the cell to prevent translation of unspliced mRNA. 58. Eukaryotic mRNA contains introns that must be removed before being translated to produce functional proteins. The splicing is done inside the nucleus before the mRNA can be accessed by ribosomes. 4. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A generic transcription factory during transcription, highlighting the possibility of transcribing more than one gene at a time. The diagram includes 8 RNA polymerases, However, the number can vary depending on cell type. The image also includes transcription factors and a porous protein core. Translation. Without the nucleus, ribosomes would translate newly transcribed, unprocessed mRNA, resulting in malformed and non-functional proteins. 1.108.15. Replication. Main article, Eukaryotic DNA replication. The main function of the cell nucleus is to control gene expression and mediate the replication of DNA during the cell cycle. 171. It has been found that replication happens in a localized way in the cell nucleus. In the S phase of interphase of the cell cycle, replication takes place. Contrary to the traditional view of moving replication forks along stagnant DNA, a concept of replication factories emerged, which means replication forks are concentrated towards some immobilized factory regions, through which the template DNA strands pass like conveyor belts. 59. Gene expression. Main article, Gene expression. See also, transcription factories. Gene expression first involves transcription, in which DNA is used as a template to produce RNA. In the case of genes encoding proteins, that RNA produced from this process is messenger RNA, mRNA, which then needs to be translated by ribosomes to form a protein. As ribosomes are located outside the nucleus, mRNA produced needs to be exported. 60. Since the nucleus is the site of transcription, it also contains a variety of proteins that either directly mediate transcription or are involved in regulating the process. These proteins include helicases, which unwind the double-stranded DNA molecule to facilitate access to it, RNA polymerases, which bind to the DNA promoter to synthesize the growing RNA molecule, topoisomerases, which change the amount of supercoiling in DNA, helping it wind and unwind, as well as a large variety of transcription factors that regulate expression. 61. Processing of premna. Main article, Post-transcriptional modification. Newly synthesized mRNA molecules are known as primary transcripts or premna. They must undergo post-transcriptional modification in the nucleus before being exported to the cytoplasm. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Macromolecules, such as RNA and proteins, are actively transported across the nuclear membrane in a process called the RAN-GTP nuclear transport cycle. mRNA that appears in the cytoplasm without these modifications is degraded rather than used for protein translation. The three main modifications are 5-capping, 3-polyadenylation, and RNA splicing. While in the nucleus, premna is associated with a variety of proteins in complexes known as heterogeneous ribonucleoprotein particles, HNRNPs. Addition of the 5-cap occurs co-transcriptionally and is the first step in post-transcriptional modification. The 3-polyadenine tail is only added after transcription is complete. 1.509.18. RNA splicing, carried out by a complex called the spliceosome, is the process by which introns, or regions of DNA that do not code for protein, are removed from the premna and the remaining exons connected to reform a single continuous molecule. This process normally occurs after 5 capping and 3 polyadenylation, but can begin before synthesis is complete in transcripts with many exons. 494 many preMNAs can be spliced in multiple ways to produce different mature MNAs that encode different protein sequences. This process is known as alternative splicing, and allows production of a large variety of proteins from a limited amount of DNA. 62. Dynamics and regulation. Nuclear transport. Main article, Nuclear transport. The entry and exit of large molecules from the nucleus is tightly controlled by the nuclear pore complexes. Although small molecules can enter the nucleus without regulation. 63. Macromolecules such as RNA and proteins require association karyophorins called importins to enter the nucleus and exportins to exit. Cargo proteins that must be translocated from the cytoplasm to the nucleus contain short amino acid sequences known as nuclear localization signals, which are bound by importins, while those transported from the nucleus to the cytoplasm carry nuclear export signals bound by exportins. The ability of importins and exportins to transport their cargo is regulated by GT parses, enzymes that hydrolyze the molecule guanosine triphosphate, GTP, to release energy. The key GT parse in nuclear transport is RAN, which is bound to either GTP or GDP, guanosine diphosphate, depending on whether it is located in the nucleus or the cytoplasm. Whereas importins depend on RAN GTP to dissociate from their cargo, exportins require RAN GTP in order to bind to their cargo. 14. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. An image of a newt lung cell stained with fluorescent dyes during metaphase. The mitotic spindle can be seen, stained green, attached to the 2 sets of chromosomes, stained light blue. All chromosomes but one are already at the metaphase plate. Nuclear import depends on the importin binding its cargo in the cytoplasm and carrying it through the nuclear pore into the nucleus. Inside the nucleus, RAN GTP acts to separate the cargo from the importin, allowing the importin to exit the nucleus and be reused. Nuclear export is similar, as the exportin binds the cargo inside the nucleus in a process facilitated by RAN GTP, exits through the nuclear pore, and separates from its cargo in the cytoplasm. 64. Specialized export proteins exist for translocation of mature mRNA and tRNA to the cytoplasm after post-transcriptional modification is complete. This quality control mechanism is important due to these molecules' central role in protein translation. Misexpression of a protein due to incomplete excision of exons or misincorporation of amino acids could have negative consequences for the cell. Thus, incompletely modified RNA that reaches the cytoplasm is degraded rather than used in translation. 1. Assembly and disassembly. During its lifetime, a nucleus may be broken down or destroyed, either in the process of cell division or as a consequence of apoptosis, the process of programmed cell death. During these events, the structural components of the nucleus, the envelope and lamina, can be systematically degraded. In most cells, the disassembly of the nuclear envelope marks the end of the prophase of mitosis. However, this disassembly of the nucleus is not a universal feature of mitosis and does not occur in all cells. Some unicellular eukaryotes, for example, yeasts, undergo so-called closed mitosis, in which the nuclear envelope remains intact. In closed mitosis, the daughter chromosomes migrate to opposite poles of the nucleus, which then divides in two. The cells of higher eukaryotes, however, usually undergo open mitosis, which is characterized by breakdown of the nuclear envelope. The daughter chromosomes then migrate to opposite poles of the mitotic spindle, and new nuclei reassemble around them. 854. At a certain point during the cell cycle in open mitosis, the cell divides to form two cells. In order for this process to be possible, each of the new daughter cells must have a full set of genes, a process requiring replication of the chromosomes as well as segregation of the separate sets. This occurs by the replicated chromosomes, the sister chromatids, attaching to microtubules, which in turn are attached to different centrosomes. The sister chromatids can then be pooled to separate locations in the cell. In many cells, the centrosome is located in the cytoplasm, outside the nucleus. The microtubules would be unable to attach to the chromatids in the presence of the nuclear envelope. 65. Therefore, the early stages in the cell cycle, beginning in prophase and until around prometaphase. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The nuclear membrane is dismantled. 18. Likewise, during the same period, the nuclear lamina is also disassembled, a process regulated by phosphorylation of the lamins by protein kinases such as the CDC2 protein kinase. 66. Towards the end of the cell cycle, the nuclear membrane is reformed, and around the same time, the nuclear lamina are reassembled by dephosphorylating the lamins. 66. However, in dinoflagellates, the nuclear envelope remains intact, the centrosomes are located in the cytoplasm, and the microtubules come in contact with chromosomes, whose centromeric regions are incorporated into the nuclear envelope, the so-called closed mitosis with extranuclear spindle. In many other protists, for example, ciliates, sporozoans, and fungi, the centrosomes are intranuclear, and their nuclear envelope also does not disassemble during cell division. 67. Apoptosis is a controlled process in which the cell's structural components are destroyed, resulting in death of the cell. Changes associated with apoptosis directly affect the nucleus and its contents, for example, in the condensation of chromatin and the disintegration of the nuclear envelope and lamina. The destruction of the lamin networks is controlled by specialized apoptotic proteases called caspases, which cleave the lamin proteins and, thus, degrade the nucleus structural integrity. Lamin cleavage is sometimes used as a laboratory indicator of caspase activity in assays for early apoptotic activity. 18. Cells that express mutant caspase-resistant lamins are deficient in nuclear changes related to apoptosis, suggesting that lamins play a role in initiating the events that lead to apoptotic degradation of the nucleus. 18. Inhibition of lamin assembly itself is an inducer of apoptosis. 68. The nuclear envelope acts as a barrier that prevents both DNA and RNA viruses from entering the nucleus. Some viruses require access to proteins inside the nucleus in order to replicate and forward/or assemble. DNA viruses, such as herpes virus, replicate and assemble in the cell nucleus, and exit by budding through the inner nuclear membrane. This process is accompanied by disassembly of the lamina on the nuclear face of the inner membrane. 18. Disease-related dynamics. Initially, it has been suspected that immunoglobulins in general and autoantibodies in particular do not enter the nucleus. Now there is a body of evidence that under pathological conditions, for example lupus erythematosus, IgG can enter the nucleus. 69. Nuclei per cell. Most eukaryotic cell types usually have a single nucleus, but some have no nuclei, while others have several. This can result from normal development, as in the maturation of mammalian red blood cells, or from faulty cell division. 70. Anucleated cells. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Human red blood cells, like those of other mammals, lack nuclei. This occurs as a normal part of the cell's development. An anucleated cell contains no nucleus and is, therefore, incapable of dividing to produce daughter cells. The best known anucleated cell is the mammalian red blood cell, or erythrocyte, which also lacks other organelles, such as mitochondria, and serves primarily as a transport vessel to ferry oxygen from the lungs to the body's tissues. Erythrocytes mature through erythropoiesis in the bone marrow, where they lose their nuclei, organelles, and ribosomes. The nucleus is expelled during the process of differentiation from an erythroblast to a reticulocyte, which is the immediate precursor of the mature erythrocyte. 71. The presence of mutagens may induce the release of some immature micronucleated erythrocytes into the bloodstream. 72. 73. Anucleated cells can also arise from flawed cell division, in which one daughter lacks a nucleus and the other has two nuclei. In flowering plants, this condition occurs in sieve tube elements. 74. Multinucleated cells. Main article, multinucleate. Multinucleated cells contain multiple nuclei. Most acantharan species of protozoa, 75, and some fungi in mycorrhizae, 76, have naturally multinucleated cells. Other examples include the intestinal parasites in the genus Giardia, which have two nuclei per cell. 77. Ciliates have two kinds of nuclei in a single cell, a somatic macronucleus and a germline micronucleus. 78. In humans, skeletal muscle cells, also called myocytes and syncytium, become multinucleated during development. The resulting arrangement of nuclei near the periphery of the cells allows maximal intracellular space for myofibrils. 1. Other multinucleate cells in the human are osteoclasts, a type of bone cell. Multinucleated and binucleated cells can also be abnormal in humans, for example, cells arising from the fusion of monocytes and macrophages, known as giant multinucleated cells, sometimes accompany inflammation. 79, and are also implicated in tumor formation. 80. A number of dinoflagellates are known to have two nuclei. Unlike other multinucleated cells, these Nuclei contain two distinct lineages of DNA, one from the dinoflagellate and the other from a symbiotic diatom. 81. Evolution. As the major defining characteristic of the eukaryotic cell, the nucleus's evolutionary origin has been the subject of much speculation. Four major hypotheses have been proposed to explain the existence of the nucleus, although none have yet earned widespread support. 82, 83, 84. The first model, known as the syntrophic model, proposes that a symbiotic relationship between the archaea and bacteria created the nucleus containing eukaryotic cell. Organisms of the archaea and explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Bacterial domains have no cell nucleus. 85. It is hypothesized that the symbiosis originated when ancient archaea similar to modern methanogenic archaea invaded and lived within bacteria similar to modern myxobacteria, eventually forming the early nucleus. This theory is analogous to the accepted theory for the origin of eukaryotic mitochondria and chloroplasts, which are thought to have developed from a similar endosymbiotic relationship between proto-eukaryotes and aerobic bacteria. 86. One possibility is that the nuclear membrane arose as a new membrane system following the origin of mitochondria in an archaebacterial host. 87. The nuclear membrane may have served to protect the genome from damaging reactive oxygen species produced by the protomitochondria. 88. The archaeal origin of the nucleus is supported by observations that archaea and eukarya have similar genes for certain proteins, including histones. Observations that myxobacteria are motile, can form multicellular complexes, and possess kinases and G proteins similar to eukarya, support a bacterial origin for the eukaryotic cell. 89. A second model proposes that proto-eukaryotic cells evolved from bacteria without an endosymbiotic stage. This model is based on the existence of modern planktomystotobacteria that possess a nuclear structure with primitive pores and other compartmentalized membrane structures. 90. A similar proposal states that a eukaryote-like cell, the chronocyte, evolved first and phagocytosed archaea and bacteria to generate the nucleus and the eukaryotic cell. 91. The most controversial model, known as viral eukaryogenesis, posits that the membrane-bound nucleus, along with other eukaryotic features, originated from the infection of a prokaryote by a virus. The suggestion is based on similarities between eukaryotes and viruses such as linear DNA strands, mRNA capping, and tight binding to proteins, analogizing histones to viral envelopes. One version of the proposal suggests that the nucleus evolved in concert with phagocytosis to form an early cellular predator. 92. Another variant proposes that eukaryotes originated from early archaea infected by poxviruses, on the basis of observed similarity between the DNA polymerases in modern poxviruses and eukaryotes. 93, 94. It has been suggested that the unresolved question of the evolution of sex could be related to the viral eukaryogenesis hypothesis. 95. A more recent proposal, the exomembrane hypothesis, suggests that the nucleus instead originated from a single ancestral cell that evolved a second exterior cell membrane, the interior membrane. Enclosing the original cell then became the nuclear membrane and evolved increasingly elaborate pore structures for passage of internally synthesized cellular components such as ribosomal subunits. 96. History. The nucleus was the first organelle to be discovered. What is most likely the oldest preserved? Drawing dates back to the early microscopist Antony van Leeuwenhoek, 1632–1723. He observed a lumen, the nucleus, in the red blood cells of salmon. 97. Unlike mammalian red blood cells, those of other vertebrates still contain nuclei. 98. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Oldest known depiction of cells and their nuclei. By Antony van Leeuwenhoek, 1719. Drawing of a chironomus salivary gland cell published by Walther Fleming in 1882. The nucleus contains polyton chromosomes. The nucleus was also described by Franz Bauer in 1804, 99, and in more detail in 1831 by Scottish botanist Robert Brown in a talk at the Linnean Society of London. Brown was studying orchids under the microscope when he observed an opaque area, which he called the areola or nucleus, in the cells of the flower's outer layer. 100. He did not suggest a potential function. In 1838, Matthias Schleiden proposed that the nucleus plays a role in generating cells, thus he introduced the name cytoblast, cell builder. He believed that he had observed new cells assembling around cytoblasts. Franz Main was a strong opponent of this view, having already described cells multiplying by division and believing that many cells would have no nuclei. The idea that cells can be generated de novo, by the cytoblast, or otherwise, contradicted work by Robert Remak, 1852, and Rudolf Virchow, 1855, who decisively propagated the new paradigm that cells are generated solely by cells, omnicellular, acellular. The function of the nucleus remained unclear. 101. Between 1877 and 1878, Oscar Hertwig published several studies on the fertilization of sea urchin eggs, showing that the nucleus of the sperm enters the oocyte and fuses with its nucleus. This was the first time it was suggested that an individual develops from a single nucleated cell. This was in contradiction to Ernst Haeckel's theory that the complete phylogeny of a species would be repeated during embryonic development, including generation of the first nucleated cell from a mononucleus, a structureless mass of primordial protoplasm, Urschleim. Therefore, the necessity of the sperm nucleus for fertilization was discussed for quite some time. However, Hertwig confirmed his observation in other animal groups, including amphibians and mollusks. Edouard Strasburger produced the same results for plants in 1884. This paved the way to assign the nucleus an important role in heredity. In 1873, August Weismann postulated the equivalence of the maternal and paternal germ cells for heredity. The function of the nucleus as carrier of genetic information became clear only later, after mitosis was discovered and the Mendelian rules were rediscovered at the beginning of the 20th century. The chromosome theory of heredity was therefore developed. 101. See also computed corpuscle sectioning, nucleus, neuroanatomy, nucleid Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Nucleomorph. References. 1 ABCDEFGHIJKLM Lodish H, Burko, Matsuda P, Kaiser CA, Krieger M, Scott MP, Zipersky SL. Darnell J, 2004. Molecular Cell Biology, 5th ed. New York, W. H. Freeman. ISBN 978-0-7167-2672-2. 2 Aaron Hofer Marie, June, 2004. Chromatin Dynamics at DNA Replication, Transcription and Repair. Review. European Journal of Biochemistry. 271. 12 2335 49 do 10.1111/J.1432 1033.2004.04162.x PMID 151823449 3 Grigory of Sat, Bulinko Popova EY, 2006. The end adjusts the means, heterochromatin. Remodeling during terminal cell differentiation. Review. Chromosome Research. 14, 15369. Do. 10.1577-005-1021-6. PMID 165060996. S2 SID 6040822. 4 Skardine M, Chromati, Hager HD, Lang M, December 1985. Specific staining of human chromosomes in Chinese hamster X-man hybrid cell lines demonstrates interphase chromosome. Territories, PDF. Primary. Human Genetics. 71, 42817 do, 10.1007/BF00388452. PMID 2416668. S2 SID 9261461. 5 Lamande I, Earnshaw W.C., April 1998. Structure and Function in the Nucleus, PDF. Review. Science. 280, 5363 54753. Site CX 10.1.1.323.5543. Do. 10.1126/science.280.5363.547. PMID 9554838. 6 Katso, Lampel S, Nikolenko J.E., Bradel J., Benio, Zerbel R.M., et al. December 1996. Active and inactive genes localize preferentially in the periphery of chromosome territories. Primary. The Journal of Cell Biology. 135, 1195 do. 10.1083/JCB.135.5.1195. PMC 21211085. PMID 8947544. Archived from the original on 29 September 2007. 7 Rothfield NF, Stiller BD, November 1967. The relation of immunoglobulin class, pattern of anti-nuclear antibody, and complement-fixing antibodies to DNA in sera from patients with systemic lupus erythematosus. Primary. The Journal of Clinical Investigation. 46, 11 1785 94. Do. 10.1172/JCI 105669. PMC 292929. PMID 416871. 8 Bond S. Goodman AD, Matson DH, February 1995. Frequency of antinuclear antibodies in multiple sclerosis. Primary neurology. 45, 23845 10.1012/WNL.45.2.384. PMID 78545544. S2 SID 30482028. 9 Coombe K, Cantwell H, Newman FR, Jones AW, Snijdes AP, Nurse P, May 2017. A systematic genomic screen implicates nucleocytoplasmic transport and membrane growth in nuclear size control. PLOS Genet. 13, 5E100677 do. 10.1371/journal.pgen.10067767. PMC 54366639. PMID 285450558. 10 ABCDEFGHIJK Alberts B, Johnson O, Lewis J, Morgan D, Raff M, Roberts K, Walter P, 2015. Molecular Biology of the Cell, 6 ed. New York, Garland Science. 11 ABC Lodish HF, Burko, Kaiser C, Krieger M, Britscher, Plo H, et al. 2016. Molecular Cell Biology, 8th ed. New York, W.H. Freeman. ISBN 978-1-4041-8339-3. 12 Shulga N, Masama Parast N, Wozniak R, Goldfarb D.S., May 2000. Yeast nucleoperins involved in passive nuclear envelope permeability. Primary. The Journal of Cell Biology. 149, 5 102738. Do. 10.1083/JCB.149.5.1027. PMC 21748828. PMID 10831607. 13 Alberts, Bruce, 2019. Essential Cell Biology, 5th ed. New York. P 242. ISBN 9780393680393. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 14 AB Pemberton LF, Pascal BM, March 2005. Mechanisms of receptor-mediated nuclear import and nuclear export. Review. Traffic. 6, 318798 do. 10.1011/J.100600-0854.2005.00270.x. PMID 15702987. S2 SID 172279. 15 ABC Alberts B, Johnson O, Lewis J, Raff M, Roberts K, Walter P, eds. 2002. Chapter 4, DNA and Chromosomes. Molecular Biology of the Cell, 4th ed. New York, Garland Science. pp. 191234. ISBN 978-0-8153-4072-0. 16-carat Stuurman N., Heinz S., Ibayu, 1998. Nuclear lamins, their structure, assembly, and interactions. Review. Journal of Structural Biology. 122, 1242-66 to 10.1006/sp.1998.3987. PMID 9724605. 17-carat Goldman A., Moir Road, Montagloe M., Stewart M., Goldman Road, November 1992. Pathway of incorporation of microinjected lamina into the nuclear envelope. Primary. The Journal of Cell Biology. 119, 472535 10.1083/JCB.119.4.725. PMC 2289687. PMID 1429833. 18-carat ABCDE Goldman Road, Gruenbaum Y., Moir Road, Shoemaker DK, Span TP, March 2002. Nuclear lamins, building blocks of nuclear architecture. Review. Genes and Development. 16, 553347. Do. 10.1101/GAD.9605002. PMID 118777373. 19-carat Broas JL, Ramakers FC, 2004. Dynamics of Nuclear Laminar Assembly and Disassembly. Review. Symposia of the Society for Experimental Biology, 56-17792. ISBN 9781134279838. PMID 1556581. 20-carat Moir Road, UNM, CUNES, Goldman Road, December 2000. Nuclear lamins A and B1, different. Pathways of assembly during nuclear envelope formation in living cells. Primary. The Journal of Cell Biology. 151, 1155-68 do. 10.1083/JCB.151.6.1155. PMC 2190592. PMID 1112142. 21-carat Span TP, Goldman A., Wang C., Huang S., Goldman Road, February 2002. Alteration of nuclear lamin. Organization inhibits RNA polymerase II-dependent transcription. Primary. The Journal of Cell Biology. 156, 46038 do. 10.1083/JCB.200112047. PMC 2174089. PMID 118543006. 22-carat Mountkeys LC, Stewart CL, June 2004. Aging and Nuclear Organization, Lamins and Progeria. Review. Current Opinion in Cell Biology. 16, 33227 do. 10.1016/J.Seb.2004.03.009. PMID 151453558. 23-carat Hernandez Verdun D., January 2006. Nucleolus, From Structure to Dynamics. Review. Histochemistry and Cell Biology. 125, 10.122737 do 10.1007/S00418-005-0046-4 PMID 163284431 S2 SID 207692660 24 AB Lamande I, Sleeman J.E., October 2003. Nuclear Substructure and Dynamics. Review. Current. Biology. 13, 21 R825-8. Bibcode, 2003 C-Bio, 13.R825L. Do. 10.1000.j.cub.2003.10.012. PMID 1458826. S2 SID 16865665. 25 Spectre DL, Lamande I, February 2011. Nuclear speckles. Review. Cold Spring Harbor. Perspectives in Biology. 3, 2000646 10.1001/perspect.0000646. PMC 3039535. PMID 209266517. 26 Alexander K.A., Kota, Nguyen SC, Zhang L., Berger SL, March 2021. 53 P mediates target gene. Association with nuclear speckles for amplified RNA expression. Primary. Molecular cell. 81, 8. S1097-2765, 21, 00174x2. 10.1016/j.molchel.2021.03.006. PMC 883037 PMID 3382340. S2 SID 233172170. 27 AB Lamande I, Spectre DL, August 2003. Nuclear speckles, a model for nuclear organelles. Review. Nature Reviews. Molecular Cell Biology. 4, 8600512 do. 10.1038/NRM 1172. PMID 1292352. S2 SID 64394113. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 28 Tripathike, Parnike VK, September 2008. Differential dynamics of splicing factor SC35 during the cell cycle, PDF. Primary. Journal of Biosciences. 33, 334554 do. 10.1007/1238-008-005-4-3. PMID 190052334. S2 SID 63329495. Archived, PDF, from the original on 15 November 2011. 29 Tripathike, Parnike VK, September 2008. Differential dynamics of splicing factor SC35 during the cell cycle. Primary. Journal of Biosciences. 33, 334554 do. 10.1007/1238-008-005-4-3. PMID 190052334. S2 SID 63329495. 30 Handwerger KE, Gall JG, January 2006. Subnuclear organelles, New Insights into Form and Function. Review. Trends in Cell Biology. 16, 1/1926 to 10.1016/J.TCB.2005.11.005. PMID 163254006. 31 cellular component nucleus speckle. UniPROT, UniPRIP. Retrieved 30 August 2013. 32 Gall JG, Bellini M, Wuz, Murphy C, December 1999. Assembly of the Nuclear Transcription and Processing Machinery, Cagal Bodies, Coiled Bodies, and Transcriptosomes. Primary Molecular Biology of the Cell. 10, 12 4385 402 do. 10.1091/mbc.10.12.4385. PMC 25765. PMID 105886665. 33 AB Matra AG, Turns RM, Turns MP, March 2007. Non-coding RNAs, Lessons from the Small Nuclear and Small Nucleolar RNAs. Review. Nature Reviews. Molecular Cell Biology. 8, 320920. Do. 10.1038/NRM 2124. PMID 173182225. S2 SID 3026805. 34 but P, Chao, Imert B, It al. May 2024. Genome Organization around Nuclear Speckles Drives. mRNA Splicing Efficiency. Nature. 629, 5 1165 1173 do. 10.1038/S41586-024-07429-6. PMC 11164319. PMID 3872076. 35 ABCCOSEM, Lamande I, 2005. Cagal Bodies, A Long History of Discovery. Review. Annual Review of Cell and Developmental Biology. 21, 10531. Do. 10.101146/Anav.cellbio.20.0104003.103738. PMID 162124889. S2 SID 8807-316. 36 ABC Pollard TD, Earnshaw WC, 2004. Cell Biology. Philadelphia, Saunders. ISBN 978-0-7216. 3362-2. 37 ABC Matra AG, Freemister, August 1998. Coiled Bodies and Gems, Janus or Gemini. Review. American Journal of Human Genetics. 63, 231721 do. 10.1086/301992. PMC 1377332. PMID 9683623. 38 Matra AG, August 1998. Of Coiled Bodies, Gems, and Salmon. Review. Journal of Cellular Biochemistry. 70, 218192 do. 10.1002 Sisi, 1097-4644, 199808001, 70:23.0.co, 2K. PMID 9671224. S2 SID 44941483. 39 Naviscus J, Bershano Mount, Tucker K.E., Lafarga M., Matra A.G., June 2004. Targeting SMN to Kagel. Bodies and nuclear gems during neuritogenesis. Primary. Chromosome 112, 8398409. Do 10.1017/00412-004-02855-5. PMC 1592132. PMID 151642113. 40 ABCD Lafarga M, Bershano Mount, Pina E, Mayo I, Castano JG, Bowman D, et al. August 2002. Clastosome, a subtype of nuclear body enriched in 19S and 20S protosomes, ubiquitin, and protein. Substrates of protosome. Primary. Molecular biology of the cell. 13, 82771-82. Site CX 10.1.1.321.6138. Do 10.1091/mbc.e02-03-0122. PMC 117941. PMID 1218135. 41 ABC Dundem, Misty Lighty, June 2001. Functional architecture in the cell nucleus. Review. The Biochemical Journal. 356, PT 2297310 do. 10.1042/026/4021:3560297. PMC 1221839. PMID 1136875. 42 Bond CS, Fox R, September 2009. Paraspeckles, nuclear bodies built on long non-coding RNA. Review. The Journal of Cell Biology. 186, 563744 do. 10.1083/JCB.2009061113. PMC 2742191. PMID 197208772. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 43 HH, Wallow I, January 1997. Nemarlin myopathy with intranuclear rods. Intranuclear rod myopathy. Review. Neuromuscular disorders. 7, 113.9 do. 10.1016/0960-8966, 96, 00404X. PMID 91321335. S2 SID 295842117. 44 Saunders W.S., Cook C.A., Earnshaw W.C., November 1991. Compartmentalization within the nucleus. Discovery of a novel subnuclear region. Primary. The Journal of Cell Biology. 115, 491931. Do. 10.1083/JCB.115.4.919. PMC 2289954. PMID 1955462. 45 Pomboa, Kulo P, Skull W, Yoon J.B., Rhoda R.G., Cook P.R., Murphy S., March 1998. Regional and temporal specialization in the nucleus, a transcriptionally active nuclear domain rich in PTF, October 1 and pica antigens associates with specific chromosomes early in the cell cycle. Primary. The EMBO. Journal. 17, 6 1768 78 do. 10.1093/17.6.1768. PMC 11700524. PMID 9501098. 46 Zimbabwe, Nguyen QD, Chespark C, October 2004. Nuclear bodies and compartments, functional roles and cellular signaling in health and disease. Review. Cellular signaling. 16, 10 1085 104. Do. 10.1016.j.cellsig.2004.03.020. PMID 15240004. 47 Lallemandbria 10 back v. De the H, May 2010. PML Nuclear Bodies. Review. Cold Spring Harbor. Perspectives in Biology. 2, 50000661 do. 10.1101/kshperspect.0000661. PMC 2857171. PMID 204529555. 48 AB Fox R, Lamande I, July 2010. Paraspeckles. Review. Cold Spring Harbor Perspectives in Biology. 2, 70000687 do. 10.1101/kshperspect.0000687. PMC 2890200. PMID 2057317. 49 AB Foxo, Bickmore W, 2004. Nuclear compartments, paraspeckles. Nuclear protein database. Archived from the original on 10 September 2008. Retrieved 6 March 2007. 50 AB Fox R, Bond CS, Lamande I, November 2005. P54 NRB forms a heterodimer with PSP1 that localizes to paraspeckles in an RNA-dependent manner. Primary. Molecular biology of the cell. 16. 11 5304 15 do. 10.1091/mbc.e05-06-0587. PMC 12666428. PMID 16148043. 51 Nakigawa S, Yamazaki T, Heroes T, October 2018. Molecular dissection of nuclear paraspeckles. Towards understanding the emerging world of the RNP milieu. Review. Open Biology. 8, 10180150. Do. 10.1098/asob.180150. PMC 62232218. PMID 3035555. 52 Pisani G, Baron B, December 2019. Nuclear paraspeckles function in mediating gene regulatory and apoptotic pathways. Review. Non-coding RNA research. 4, 41283134. Do. 10.1016/j.nkana.2019.11.002. PMC 7012776. PMID 32072080. 53 Kong XN, Yang HX, Chen L, Dong LW, Yang W, Lu Q, It AL. October 2007. LPS-induced down regulation of signal regulatory protein, alpha, contributes to innate immune activation in macrophages. Primary. The Journal of Experimental Medicine. 204, 11 2719 31 do. 10.1084/gem.20062611. PMC 21184889. PMID 179545668. 54 AB Carmo Fonseca M, Bershino Mount, Lafarga M, September 2010. Orphan Nuclear Bodies. Review. Cold Spring Harbor Perspectives in Biology. 2, 9000703 do. 10.1011/perspect.0000703. PMC 2926751. PMID 206105447. 55 Sampuda Kilometer, Riley M., Boyd L., April 2017. Stress-induced nuclear granules form in response to accumulation of misfolded proteins in Caenorhabditis elegans. Primary. BMC Cell Biology. 18, 118. do. 10.101186/017-01360X. PMC 5395811. PMID 284240553. 56 Learninger L., Nelson D. L., Cox mm, 2000. Learninger Principles of Biochemistry, 3rd ed. New York. Worth Publishers. ISBN 978-1-57259-931-4. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 57 Moreno F., Awat CD, Rio, Palomino CA, Hariro P., February 2005. Glucose sensing through the HXK2 dependent signaling pathway. Primary. Biochemical Society Transactions. 33, PT 12658. do. 10.1042/BST03302665. PMID 156673222. S2 SID 206470022. 58 Gorlick D., Kitayu, 1999. Transport between the cell nucleus and the cytoplasm. Review. Annual Review of Cell and Developmental Biology. 15, 160760 do. 10.1014/anav.cellbio.15.1.607. PMID 106119774. 59 Hazark P., Cook PR, February 1994. Replication Factories. Review. Trends in Cell Biology. 4, 248. 52 do. 10.1016/0962-8924, 94, 90009-4. PMID 147318666. 60 Nyhors KH, Wilson DN, 2004. Protein Synthesis and Ribosome Structure, translating the genome. Huila VCH. ISBN 978-3-527-30638-1. 61 Nicolini CA, 1997. Genome Structure and Function, From Chromosomes Characterization to Genes. Technology. Springer. ISBN 978-0-7923-4565-7. 62 Black DL, 2003. Mechanisms of Alternative Pre-Messenger RNA Splicing, PDF. Review. Annual Review of Biochemistry. 72, 1291336 10.1146-2.biochem.72.1218001.1617120. PMID 126266338. S2 SID 235762888. 63 Watson J.D., Baker Ta, Bell S.P., Gano, Levine M., Lozic R., 2004. Ch 910. Molecular Biology of the Gene, 5th ed. Peason Benjamin Cummings, CSHL Press. ISBN 978-0-8053-9603-4. 64 Cavers R.T., Vernos I., 2015. The RAN-GTP pathway, from nucleocytoplasmic transport to spindle assembly and beyond. Review. Frontiers in Cell and Developmental Biology. 3, 82. Do. 10.3089/FSL.2015.00082. PMC 4707252. PMID 267933706. 65 Lippincott Schwartz J., March, 2002. Cell Biology, Ripping Up the Nuclear Envelope. Commentary. Nature. 416, 6876 31-2. Bibcode, 2002 Nature.416, 31L. Do. 10.1038/4160318. PMID 11882878. S2 SID 443100. 66 AB Bolicas T, 1995. Phosphorylation of transcription factors and control of the cell cycle. Review. Critical reviews in eukaryotic gene expression. 5, 1177. PMID 7549180. 67 Beecher B, Barrel Y, 2013. The Cell Biology of Open and Closed Mitosis. Review. Nucleus. 4, 3. Austin, Tex, 165. Do. 10.4061/nucal.24676. PMC 372075. PMID 2364439. 68 Steen R.L., Calas P., April, 2001. Mr. Getting of B-type lamins at the end of mitosis, implications on cell survival and regulation of lamins a /C expression. Primary. The Journal of Cell Biology. 153, 3. 6216. Do. 10.1038/JCB.153.3.621. PMC 21905667. PMID 11331311. 69 Bomai, November, 2007. IgG deposits can be detected in cell nuclei of patients with both lupus erythematosus and malignancy. Primary clinical rheumatology. 26, 11 1877 82 do. 10.1007/S10067 007-0597Y PMID 1736435 S2 SID 44879431 70 Ressel L. 2017. Nuclear morphologies. Normal cell morphology in canine and feline cytology, an identification guide. Hoboken, N.J., John Wiley and Sons. P6. ISBN 978-1-119-27891-7. 71 Skutelski E., Dannon D., June, 1970. Comparative study of nuclear expulsion from the late erythroblast and cytokinesis. Primary experimental cell research. 60, 342736 do. 10.1016/0014 4827, 70, 90536-7 PMID 5422968 72 Taras DK, Dertinger SD, Hall NE, Tometsko CR, February 2000. Enumeration of micronucleated reticulocytes in rat peripheral blood, a flow cytometric study. Primary mutation research. 465, 1291. 9 do. 10.1016/S1383-5718, 99, 00216-8. PMID 10708974. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 73 Hutter KJ, Stoer M, 1982. Rapid detection of mutagen-induced micronucleated erythrocytes by flow cytometry. Primary histochemistry. 75, 335362 do. 10.1007/BF00496738. PMID 71418888. S2 SID 28973947. 74 Ham BK, Lucas WJ, April 2014. The angiosperm flow emissive tube system, a role in mediating trays. Important to modern agriculture. Journal of Experimental Botany. 65, 7 1799 816. Do. 10.1093/JXB/ERT417. PMID 243685003. 75 Zettler LA, Sogin ML, Caron the, October 1997. Phylogenetic relationships between the Acanthera and the Polycystinia, a molecular perspective on Haeckel's radiolaria. Primary proceedings of the National Academy of Sciences of the United States of America. 94, 21114116. Bibcode, 1997 NAS, 9411410. Do. 10.1073/NAS.94.21.11411. PMC 23483. PMID 9326623. 76 Horton TR, 2006. The number of nuclei in basidiospores of 63 species of ectomycorrhizal Homobasidiomystes. Primary. Mycologia. 98, 22338 10.3852/mycologia.98.2.233. PMID 16894968. 77 Adam Road, December 1991. The Biology of Giardia SPP. Review. Microbiological Reviews. 55, 4. 70632. Do. 10.101128/MMBR.55.4.706-732.1991. PMC 372844. PMID 17799932. 78 Vogter, Goldman AD, Moshitsuki K., Landweber LF, 1 August 2013. Transposon domestication versus mutualism in ciliate genome rearrangements. PLOS Genetics. 9, 80100365. Do. 10.1371/journal.pgen.100365. PMC 37312111. PMID 239355529. 79 McKinney-Zo, Rennick DM, February 1988. Interleukin 4 induces cultured monocytes /macrophages to form giant multinucleated cells. Primary. The Journal of Experimental Medicine. 167, 2598611. Do. 10.1084/gem.167.2.598. PMC 218885. PMID 325808. 80 Goldring SR, Roelk MS, Peterson KK, Barney K., February 1987. Human giant cell tumors of bone. Identification and characterization of cell types. Primary. The Journal of Clinical Investigation. 79, 2. 48391. Do. 10.1172/JCI112838. PMC 424109. PMID 3027126. 81 Imanian B, Gumert JF, Doral RG, Berkey F, Keeling PJ, 2012. Tertiary endosymbiosis in 2 dinotoms has generated little change in the mitochondrial genomes of their dinoflagellate hosts and diatom endosymbionts. Primary. PLOS 1. 7, 843763. Bibcode, 2012 PLOSO, 743763I. Do. 10.1371/journal.pone.0043763. PMC 342334. PMID 229163003. 82 Penise IE, August 2004. Evolutionary Biology. The Birth of the Nucleus. News. Science. 305, 5685. 7668. Do. 10.1126/science.305.5685.766. PMID 15297641. S2 SID 8376925. 83 DeVos DP, Graph Field MC, June 2014. Evolution of the Nucleus. Review. Current opinion in cell biology. 28, 108-15 do. 10.1016/j.seb.2014.01.004 PMC 4071446 PMID 245089884 84 Lopez Garcia P. Moreira D. November 2015. Open questions on the origin of eukaryotes. Review. Trends in ecology and evolution. 30, 11697708 do. 10.1016/j.3.2015.09.005 PMC 4640172 PMID 264557774 85 Hogan centimeter 2010 Archaea. In Moneson E. Cleveland C. eds. Encyclopedia of Earth. Washington, D.C., National Council for Science and the Environment. Archived from the original on 11 May 2011. 86 Margulies L. 1981. Symbiosis in Cell Evolution. San Francisco, W. H. Freeman and Company. pp. 206227. ISBN 978-0-7167-1256-5. 87 Martin W., December 2005. Archaebacteria, Archaea, and the Origin of the Eukaryotic Nucleus. Car. Appin Microbiol. 8, 6637 do. 10.1016/j.mib.2005.10.004 PMID 162429992. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 88 Bernstein, H. Bernstein, C. 2017. Sexual Communication in Archaea, the Precursor to Eukaryotic Meiosis. In Whitson, G. eds. Biocommunication of Archaea. Springer, Cham. https://do.org/10.1007/978 3-319-65536-9_7. 89 Lopez Garcia P. Moreira D. May 2006. Selective Forces for the Origin of the Eukaryotic Nucleus. Review. Bioessays. 28, 552533 do. 10.1002/bis.204113. PMID 166155090. 90 Fuest J. 2005. Intracellular compartmentation in Planktomystes. Review. Annual Review of Microbiology. 59, 299328. do. 10.1046/anov.micro.59.0308004.12128. PMID 1591029. 91 Hartman H. Fedorova, February 2002. The Origin of the Eukaryotic Cell, a Genomic Investigation. Primary. Proceedings of the National Academy of Sciences of the United States of America. 99, 3. 1425. Bibcode, 2002 NAS, 99.1420h. do. 10.1073/NAS.0326585999. PMC 122206. PMID 11805300. 92 Bell PJ, September 2001. Viral eukaryogenesis was the ancestor of the nucleus, a complex DNA virus. Comment. Journal of Molecular Evolution. 53, 32516. Bibcode 2001 JML 53251L. Do 10.1007/S0023900100215. PMID 11523012. S2 SID 20542871. 93 Takes Mura M, May 2001. Poxviruses and the Origin of the Eukaryotic Nucleus. Primary Journal of Molecular Evolution. 52, 541925. Bibcode 2001 JML 52419T. Do 10.1007/S0023900100171. PMID 11443345. S2 SID 21200827. 94 Villarreal LP, De Philippis VR, August 2000. A Hypothesis for DNA Viruses as the Origin of Eukaryotic Replication Proteins. Primary Journal of Virology. 74, 15 707984 do. 10.1128/JVI.74.15.7079. 7084.2000. PMC 112226. PMID 10888648. 95 Bell PJ, November 2006. Sex and the eukaryotic cell cycle is consistent with a viral ancestry for the eukaryotic nucleus. Primary Journal of Theoretical Biology. 243, 15463. Bibcode 2006 JB.243, 54B. Do 10.1016/J.B.2006.05.015. PMID 16846615. 96 de Roos AD, 2006. The Origin of the Eukaryotic Cell based on Conservation of Existing Interfaces. Primary Artificial Life. 12. 451323 do 10.1162/Artel.2006.12.4.513. PMID 16953783. S2 SID 5963228. 97 Van Leeuwenhoek Opera Omnia, Su Arcana Natrio Exactissimorum Microscopiarum Detector. Experimentis Veritas Comprobator, Epistolis ad varios illustres viros J. Arnold at Delphis, Abiman. Lugdinum Bataverum, the works of, or arcana of nature by means of exactissimorum microscopes had been detected and confirmed by a variety of experiments, the epistles to the various illustrious men of Vela J. Arnold and Delphi, Abiman, Lugdana York 1719–1730, in Latin. Cited in Gelac D. 2009. Geschichte der Mikroskopie. Frankfurter Main, Germany, Verlag Harry Deutsch. ISBN 978-3-8171. 1781-9. 98 Cohen W.D., 1982. The cytomorphic system of anucleate non-mammalian erythrocytes. Protoplasma. 113, 2332. Do. 10.1007/BF01283036. S2 SID 41287948. 99 Harris H., 1999. The Birth of the Cell. New Haven, Yale University Press. ISBN 978-0-300-07384-3. 100 Brown R., 1866. On the Organs and Mode of Fecundation of Orchidex and Asclepiadae. Miscellaneous Botanical Works I, 511514. 101 A.B. Chromati, 1985. Von der Zellenlehre Chromosomentheorie. Berlin, Heidelberg, New York. Tokyo, Springer Verlag. ISBN 978-3-540-13987-4. Online version here. Further reading. Goldman Road, Gruenbaum Y., Mwa Road, Shoemaker D.K., Span T.P., March 2002. Nuclear lamins, Building. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Blocks of Nuclear Architecture. Genes and Development. 16, 553347 do. 10.1101/GAD.9605002. PMID 118773773. A review article about nuclear lamins, explaining their structure and various roles. Gorlick D., Kitae U., 1999. Transport between the cell nucleus and the cytoplasm. Annual Review of Cell and Developmental Biology. 15, 60760. Do. 10.1046/anav.cellbio.15.1.607. PMID 106119774. A review article about nuclear transport, explains the principles of the mechanism, and the various transport pathways. Lamin I, Earnshaw W.C., April 1998. Structure and Function in the Nucleus, PDF. Science. 280. 5363 54753. Site CX 10.1.1.323.5543. Do. 10.1126/science.280.5363.547. PMID 9554838. A review article about the nucleus, explaining the structure of chromosomes within the organelle, and describing the nucleolus and other subnuclear bodies. Pennies IE, August 2004. Evolutionary Biology. The Birth of the Nucleus. Science. 305, 5685 7668. Do. 10.1126/science.305.5685.766. PMID 152976441. S2 SID 83769250. A review article about the evolution of the nucleus, explaining a number of different theories. Pollard T.D., Earnshaw W.C., 2004. Cell Biology. Philadelphia, Saunders. ISBN 978-0-7216-3360-2. A university-level textbook focusing on cell biology. Contains information on nucleus structure and function, including nuclear transport and subnuclear domains. External links. The nucleus. MB Info. Learn about the cell nucleus. Cellnucleus.com. Website covering structure and function of the nucleus from the Department of Oncology at the University of Alberta. Bigmore W. The Nuclear Protein Database. Medical Research Council Human Genetics Unit. Information on nuclear components. The Nucleus Collection. Image and Video Library. The American Society for Cell Biology. Archived from the original on the 12th of November, 2006. Contains peer-reviewed still images and video clips that illustrate the nucleus. Ball J.G., McIntosh Jr., eds. Nuclear Envelope and Nuclear Import Section. Landmark Papers in Cell Biology. Archived from the original on the 17th of November, 2006. Contains digitized commentaries and links to seminal research papers on the nucleus. Published online in the Image and Video Library, archived the 10th of June, 2011 at the Wayback Machine of the American Society for Cell Biology. Cytoplasmic patterns generated by human antibodies. Antibodypatterns.com. Archived from the original on the 2nd of January, 2007. Categories: cell nucleus, cell anatomy, organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.